This procedure begins by positioning the pipette tip near the polishing filament at 100 times magnification. Next, turn on air pressure and heat the filament. The pipette tip will expand to form a blunt-shaped tip. Turn off the heat. To obtain a smaller tip opening, turn off the pressure and heat again.
Hi, I'm Brandon Johnson from the laboratory of Dr. Miriam Goodman in the Department of Molecular and Cellular Physiology at Stanford University. Today, we will show you a procedure for pressure polishing patch pipettes. We use this procedure in our laboratory to study ion channels in membrane patches and in C. elegans neurons. So, let's get started.
Pull a set of 6 to 10 patch pipettes. Consult a local expert or our visualized experiment making patch pipettes and sharp electrodes with a programmable puller for help. To begin the procedure, set a compressed air line pressure to about 40 psi. Then connect the compressed air line to a 4-way control valve. Connect a length of plastic tubing to the 4-way valve and attach a high-pressure pipette holder to the loose end of the plastic tubing. Use a microforge rig to perform the polishing. We use the CPM2 kit from ALA Scientific. This rig has a high-pressure pipette holder, a platinum heating filament controlled by a foot pedal, and a microscope equipped with a low and high magnification. 10 times and 100 times long working